So I'm back. I'm going to be moderating this next panel, which is even more compressed than our prior panel in some respects, in that we have more speakers. On the other hand, we also have more time. This panel will look at new approaches to drug development. Every person on this panel has been directly involved on the ground with new approaches and thinking about new approaches, and also paths to market for those new approaches. So I think that in terms of giving us real-world insights into these questions, this panel will be extremely useful. Because I'm also the tech person, in addition to being the moderator, I'm going to go there for just a moment and set up Chuck's and Christine's slides.
Thanks, Arti, and thanks everyone for coming. This is a very important subject here today, and it affects many of the programs we have ongoing at the NIH, particularly at the National Centers for Advancing Translational Sciences. Christine, Colvis, and I will be speaking on some of our programs. So first, of course, the standard model is might consider a static model where the research ends into translation and eventually public health comes out the other end. And the way we look at it is that public health is in the center, and that all parties should have a direct opportunity to add to the value of their particular piece of the puzzle. And of course, as we've heard today already, we all know that there's been a crisis in the fundamental science. It's tremendously advanced. That's what the public hears in the newspapers, but on the other hand, people seem to be getting more unhealthy. And at the congressional level, the public and private enterprise seems to be failing, and the sources of funding are getting impatient. So we look at the drug development system in crisis, clinical trials in crisis, and just poor adoption across the board. What can we do about that? At NCATS, our mission is specifically to catalyze the generation of innovative methods and technologies that will enhance the development and testing of information of diagnostics and therapeutics across a wide range of diseases. So we're not there to develop new products, new therapeutics directly. Our job is to fill the gaps. So if there's a new way that is needed to deliver a drug to a child with a neurological disorder, and we need to get through the cranium, there's no direct market for that other than maybe if we can get involved and bring together the players who want to solve that problem. And we do this by catalyzing collaborations across various levels. So within the NIH, we have many collaborations ongoing, as well as outside the NIH. Many of you in this room collaborate with NCATS, and we try to complement and not compete with others' work. We try to revolutionize the processes again and find new ways to galvanize this, and that's primarily through partnerships. Most of what we do is all collaborative. So even though we have a strong intramural focus, which is only 10% of our budget, by the way, but it is considered intramural, 90% of that effort is collaborative outside of our group. And also we try to interact with the nonprofits and the regulatory experts, as well as develop new relationships with pharma to help them with their challenges. So some of the characteristics are that we try to address bottlenecks. We look for bottlenecks that hold up everyone and see if there's a way that we can add to the value there and break those down and open things up. Again, highly collaborative. 90% of everything we do is collaboration with the outside. So this might bring in issues of buy-dole in some cases, but in others it gives us the advantage to be able to collaborate with domain experts and to solve their problems. And in doing this is a very highly incentivized inside our group, and we try to respond quickly to these problems. So we have the division of preclinical innovation, which is considered sort of the intramural program. Like I said, 90% of that is collaborative with the outside world. We also have a division of clinical innovation I'll mention at the end, which is 90% of our budget, which involves the Clinical Translational Science Award centers across all the United States, where we try to leverage on the clinical end new innovations. So within the division of preclinical innovation, we have currently 5 major programs, but we continually try to recycle in new programs and modify the programs that are existing. So we have the TREND, the Therapeutics for Rare and Neglected Diseases program. We have Tox21, looking at trying to set up toxicology models that can be useful for everyone. Bridges, trying to bridge interventional development gaps in particular parties' technologies and IP, and Molecular Libraries program, which also includes a very important assay development group. And another unique aspect of NCATS, our DPI staff is primarily from the outside world. So we have experts from all types of small pharma, big pharma, across management as well as technical, of course, and operations. And this cannot be understated how revolutionary this is for the government to have a group that is composed of such an outside view of the world. It is, and one of the things about it is that there's amazingly no tenure at NCATS, which some people find hard to believe. Tenure is often said to be, you need tenure in order to encourage to retain the best and brightest. Actually, we're finding that the best and brightest are coming to us to work with us because we have such an innovative model across the board, whether it's in the science or whether in the agreement side or the administrative side or the funding side. We're trying to be innovative and find new ways to make it all happen. So it's actually working great, and the top-notch people we have, they could probably end up anywhere, but they choose to be with us. Again, all collaborations. The projects are selected generally by solicitation and review. We try to bridge the gaps again and be an adaptive function with other people's problems. It's disease agnostic, and we look for to try to break through common mechanisms that were previously undiscovered. And so here's sort of a layout of, you have at the top the biopharmaceutical pipeline that we're familiar with. In the center there are all our various programs. So what we try to do is we try to leverage our capabilities to solve problems. And again, this is all collaboratively. So we have people come to us with an idea for solving a problem, and we have various programs that will address that. Again, the NCGC, National Chemics Genomics Center, was initially started at the NIH in 2004. We now have over 400,000 compounds in our library, somewhere around 3,500 pharmaceutical compounds. So this gets to the repurposing question of when we get a new assay in, we do a library scan, high throughput screening scan, and identify potential targets and leads. And this program is actually being retooled. We're trying to update it and make it more amenable to IP considerations that we had problems with in the original NCGC program and publication. And again, the NCGC, the pharmaceutical collection continues to grow. The number, this is our old numbers already, but we're close to 3,500 total in our collection, and there's still a few more to go. And I'll be getting to how that, so here's our pipeline. For a new chemical entity, right, you put in 300,000 compounds, you might get one drug after 10 to 15 years. So of course with repurposing and with the screening, if we have a hit coming out of the pharmaceutical library of 3,000 drugs, we're hoping that instead of 15 years now, it'll take only 3 years. So we've starting to have some success with that. I think Aranophen is one example, and I think we may be hearing about that later today. Tox21 program I mentioned, the goal is to identify mechanisms of toxicology and serve as a template for being able to scan and do toxicity testing quickly. Bridges program, this is where people come to us with their IP, so it might be a small startup company. They can apply for Bridges funding. We have proposals that go out just about every year. And there, they have the IP. We don't add to the IP, but they have a particular problem that might be able to solve, whether it's toxicology or doing PK. We'll have a group of experts gather, and we'll serve as advisors. And we'll go through and identify problems, and we'll do this for all the applicants. And then we triage it to see who's most likely needs the money most to be able to get to that next stage. If we had more money, we'd be able to give more grants out. But the idea here is just to advance the technology one step. It's not necessarily to get a therapeutic that day, but just to be able to get you to the IND filing stage. And it's been successful thus far. Trend is a focus on rare and neglected diseases, and there we're more collaborative. It's more of a developmental collaboration, and there generally joint IP is developed because we're still trying to solve problems. And it too has been successful. Comparing the two, bridges again is we're just trying to help someone else from the outside bridge their gaps. And Trend, we're more involved in solving bigger problems and trying to get it into a clinical trial support. We also have the tissue chip screening program, and that is in collaboration with NIH, with DARPA, and FDA. And so far we've hit all the milestones on that, so we're looking forward to where this could be help with, of course, with FDA clinical trials, which are so expensive. If we can have something like this bring down the cost of early phase clinical trials, we'll be able to not only screen more compounds, but bring the cost down of drug development. And Christine will talk about the new therapeutic uses program, where we are doing drug repurposing directly. Again, the clinical and translational science awards, this is 90% of our budget, and just those who are interested, in June 2013, the Institute of Medicine released its vision report for the TSA program and how we can use that to keep up with the changing translational landscape, which touches on a lot of the points that are being brought up here today. And it's spread out all over. So again, we have this capability spread out. We have experts willing to collaborate, a little bit of funding to do it. And I guess this is my main slide that most of you, what we heard about market exclusivity. So on the left side there, we've got these various hurdles, whether it's in science or IP, meeting an unmet market need, making sure we have buy-in of patients, doctors, and payors, making sure that there is a regulatory path and that there's a known cost that can be invested in, and that there's a payer there out willing to reimburse it. So these are things that we consider every day at NCATS because these are our collaborators. And at every step, the translational advance that we try to achieve, we have to consider, well, where are we going? It's like we're building a collaborative runner here. And on the right side, what we're trying to achieve is the reward at the end of the finish line. There has to be some sort of exclusivity, and that would be, as we heard, there's obviously patent exclusivities and the FDA Hatch-Waxman type exclusivities or other FDA exclusivities that we would like to know that's there. So while a project is ongoing, if we don't see one of those out there, we're in trouble. We're going to say, well, what can we do? Should we bother investing in it? And sometimes the answer is no. And so we try to figure out, well, what else is there? So there are other aspects. So in case of, if something is off patent and there's a generic, the threat of off-label use is huge. If we develop this and we want a partner to invest in it, they're not going to invest in it if it can be used off-label. From the government's perspective, we probably see that as fine, but if there's an added investment we need from the outside collaborator to get it through clinical trials, then that collaboration won't happen. So what we're finding, actually, we have one case where we have an off-label, we have a generic compound, cyclodextrin, that's been used for many years. It's now been found to be useful for treating a rare neurological disorder, Niemann-Pick type C disease, and it's actually in clinical trials now at the NIH. The amazing thing is we're getting ready to partner that up and have investors come in, but it's off patent. There's no existing patent on the existing product, and it could potentially be, it is generic in the market. The issue is that the advantage we have is that the cyclodextrin needs to be delivered directly to the brain, so either intracranial or intrathecally, and that's not something that an off-label use would apply, right? A doctor's not going to say, go ahead, here, take this and inject this into your child's brain. You need to have a certain formulation. You need to have this, the proper clinical protocol. So there we have a leverage there to have, even though there's no IP and no exclusivity, well, there is going to be exclusivity because now we're going to have the orphan, pediatric, and breakthrough. So, but if we didn't have the unique factor of this unique formulation need to be delivered to the brain, those FDA exclusivities would not be of any value either. The other aspect I want to touch on before I finish here is data itself. We're finding that the data packages that we're able to help create with our partners in the early clinical trials has great value, as those who are in the clinical regulatory game know. And we're able to package that. We have instances where there is no IP, but we're generating this wonderful data to try to make a valuable drug. That data is hugely important to potential partners. They can take that, and not only will then validate the FDA regulatory filings, but it could potentially validate secondary filings for either related diseases or other neurological disorders that have a broader focus, such as Alzheimer's or Parkinson's. So anyway, I just want to touch on that there are other types of exclusivities, and that we really look at that every day in every program, that in order to have a path forward, we have to have a reason to cross the finish line. But on the other hand, having this collaborative stakeholder model, we've been able to increase the opportunity to overcome the risks. It's just now we need to have a reason for crossing the finish line. And so that's the challenges we face. So thank you very much.
Colvis is also with NCATS, and she's going to speak on a repurposing and rescue program.
Yeah, so thanks, Arti, and thanks for the invitation to come and talk to everybody here today. So I'm going to talk about the NIH Industry Pilot Program that we affectionately call the New Therapeutic Uses Program, which is it was a pilot that was really born out of a meeting and follow-up discussions between Francis Collins and some key players in large pharmaceutical companies. And through those discussions, it became very apparent that the time was really right for us to do something more proactive about facilitating collaborations between the pharmaceutical industry and particularly academia. And that in doing so, we might really be able to better leverage the intellectual power that exists if we go out to the broader research community versus just relying on the, what you have with the capacity that you have within a pharmaceutical company. So, so the program, for each project, of course, they're going to be looking to come up with new therapeutic uses for proprietary compounds. And as Chuck said, we are disease agnostic at NCATS, so this could be in any disease area versus if we were from NCI or from National Institute of Mental Health or something where we have a disease focus. So it could really be in any disease area. It's important to understand that for this program, none of these were FDA-approved drugs. All of the things that we are looking at are investigational drugs. And so the idea, as I said, would be, trying to match these assets that exist in the pharmaceutical industry that have been very, very well developed, that represent a huge investment on their part, to try to match that up with ideas that are coming from academia. So you have three main parties here. We have the NIH, that would be us, particularly NCATS, that worked with each of our pharmaceutical partners, each of our eight companies, to develop template agreements, and I'll say a little bit more about that at the end of my presentation, as well as, of course, we provided the funding opportunity announcements, we provided the peer review of the scientific ideas. We are providing the funding to the investigators who come up with the ideas. We are not providing any funding to the pharmaceutical companies. And because it's our money, we are also responsible for oversight of the projects. And then, of course, the pharmaceutical partner in this is bringing compounds or biologics, although for the ones that all went forward, it was all involving small molecules. They've provided a tremendous amount of in-kind support, actually, in terms of scientific support as well as regulatory support. And they, because of all the work that they've already done on these compounds, they have a tremendous wealth of data on these compounds that would be needed then in order to update the IND so that our investigators could actually move forward with them. And then we turn to the academic researchers who are going to have a much deeper knowledge. In some cases, I think of it as almost sort of a niche knowledge about a disease, and have some hopefully novel and new ideas for how to use these very well developed compounds. So that three-way relationship looks something like this. So we have a memorandum of understanding in place with each of the 8 partners, and the 8 partners are listed there on the right alphabetically. And then for every project that goes forward, there's a collaborative research agreement between the industry partner and the academic researcher. And as I said, I'll say a little bit more about those collaborative research agreements, those template agreements that we had developed later in the presentation. Our investigators all had to propose to do at least a phase 2A trial. And if they needed it, they could also propose to do a phase 1 or maybe some preclinical studies, but they all had to propose a phase 2. So the expectation here is that these are compounds that are very, very well developed. They've already been through safety trials, and they really should be ready to go into humans. We don't really need to do a whole lot of basic science research. So this is a little bit different for what the NIH does in terms of the vast majority of the stuff that we support. And so when we issued the announcement that said we have this opportunity for funding, we had to, of course, if we're going to try to get ideas from the research community, we have to tell them what compounds are actually available. So simultaneously, when we make the announcement of the funding opportunity, we also provided some information about these compounds, because again, these were proprietary compounds for the most part. These are not FDA-approved drugs. So many of them were not in the public domain in a really big way. Some of them had publications, they had clinical trial information in clinicaltrials.gov, but these weren't things that were necessarily really broadly known. So this is just sort of the top piece of the table that we published when we published the funding opportunity announcement. And so this first column here then has the compound identifier, and the second column there has the mechanism of action for what that compound actually does. Third is what the original indication was that the company was developing the compound for, and then the route of administration was listed last. And I should say too that these compounds were, in general, they were discontinued because either they failed to show efficacy when they got into patients, into a patient population, or because they were just deprioritized for business reasons. So they generally have very reasonable safety profiles when we look at that. And so this would allow an investigator to look very quickly, primarily probably focusing on the mechanism of action, see if there's anything there that looks like it would be of interest for the disease that they work on. So if they are aware that this compound or that this target may be important for the disease that they study, they could then click on the number. These numbers were all hyperlinked, and then they could click on that and they could get just a little bit more information about the compound. Probably primarily focusing on what the safety profile looks like and whether or not it looks like that would coincide well, work well with what their indication is that they would be interested in, but also looking at what the company, just a little sneak peek of what it is that the company knows about the compound from when they were developing the compound and from when they were testing the compound. So it's important to understand that at this point in the whole process, this information isn't intended to be all-encompassing. This is intended to provide just enough information for an investigator to be able to say, yeah, I think this might be a reasonable compound for treating the disease that I'm interested in. And with this information then, they would submit a very short pre-application to us. This wasn't a request for money. It wasn't a full-blown application that describes what their clinical protocol is going to be. It was just to say, here's how I think we might be able to use this compound. Here's the team that we have in place. And here's why we think that this disease, why there's still a need to have therapeutics for this particular disease. So those are the things that we're really looking at in that pre-application phase. And so when we did this, what we expected investigators to do, as I said, was they would be looking at our table of the 58 different compounds, and someone would look at a mechanism of action and come up with an idea for a use for that compound. And then this would happen for many, many compounds, coming up with all different ideas. This was the expectation that this is what would happen, and it did happen. But then the other thing that happened that we also sort of expected, but not quite to the degree that we saw it, and I think this is where we see and demonstrate the power of crowdsourcing, the power of going out to a broader research community, which was that you have this person who looks at this mechanism of action and comes up with an idea for a new use. You have other people that look at that same mechanism of action and come up with completely different ideas for the mechanism of use. And you can tell they're totally different because their light bulbs are totally different colors. And so they have these new ideas. And we know that this will happen because that is kind of the idea behind when people repurpose a drug that's an already approved drug. That is the idea. The thing that we didn't expect was the degree to which this happened. And so what I've plotted here, and this is real data from those pre-applications that we received. We got almost 160 applications with an announcement being on the street for only about 60 days. 160 ideas with 58 compounds in the offering. So these are the 16 most popular. So what I did was, if I plotted if there were 5 applications or more for the compound, it made it onto the chart, and then the different colors just represent different indications for a particular compound. And so you see that only one of the compounds that had 5 applications had 5 applications that were all kind of for the same general disease area. And everybody else came up with at least 3 different and very, very different ideas for how they might use these compounds. This to me was really, really fantastic. This to me said that even if we can't do this in a crazy fast way, we are really going to get new and different ideas by going out to the broader research community. This was incredibly cool. I cannot tell you I can't give you the real indications that were provided, but I wanted to give you a sense of what the diversity really looked like. So to sort of assure you that if we had 3 applications and one was for colon cancer and one was for breast cancer and one was for prostate cancer, I didn't consider those different diseases. I considered them solid tumor cancers, and I just bundled them all together. Just don't tell my NCI colleagues that that's what I did. But the diversity was truly, truly incredible. For one of the compounds, for example, one of the indications was for treating herpes and Alzheimer's disease. So this is kind of the breadth that we're talking about. So it was really quite incredible. So this is at the pre-application stage for these applications, for this process. And at this point then, these underwent a peer review process, and we selected the ones that were sort of at the top tier. And remember, so here we aren't looking at a full-blown application. We are looking for, is the association that you are making, the claim that you're making, that this target, and therefore this compound, might actually be useful for this indication, is that a pretty solid link? Do you have the right team put together, and you're going to be able to hit the ground running because we're only going to give you 3 years maximum to get these studies done and to finish your phase 2A trial. And is this an unmet medical need? And so we're really looking at that at this point. We take those top-tier investigators, we put them in touch with the pharmaceutical companies, and they quickly sign a confidential disclosure agreement, they swap data back and forth, and try to make a decision about, now that the investigator can see more data from the company, and the company can have more of a discussion with the PI, is it going to be worth us pursuing? Based on what we're looking at, on the whole, does the company have the capacity to take on multiple projects? And so one of the things to appreciate here is that we are sending to a single company many investigators simultaneously. They had 11 weeks to come up with their project, with their application to submit to us, which had a much more detailed description of what it was that they were proposing to do. They also had, in that 11-week period, they had to get their collaborative research agreement in place between the academic investigator, the academic institution, and the pharmaceutical company. The only way that that would have been possible, and 11 weeks, and we threw Thanksgiving in there for good measure, the only way that that was going to happen was if we had template agreements that could be pre-reviewed by the academic institutions to decide, before you even send us your pre-application, take a look at the template agreements. If you don't think you can use this as a starting point for negotiations, you probably shouldn't even send us an application, because this is where the companies are going to start. They cannot be negotiating many collaborative research agreements with multiple investigators in an 11-week period. It just wouldn't be feasible, as many people in the room here would know. So these template agreements are really what made the crowdsourcing possible. The companies would never have engaged with us if we weren't able to do this. We, the NIH, negotiated on behalf of our investigator community. We pre-negotiated these with our companies. We made them publicly available. They're still publicly available. And as I mentioned, then these served as the starting point for negotiations. And that is, so then in June, just this past June, is when we issued awards. We got 9 awards going, covered 8 different disease areas. We were very pleased with the diversity of the diseases that we actually were able to cover. And we have already, I think 3 of them are already dosing patients. We're dosing patients as of August. So just within a couple of months of making the awards, people are ready. And by the end of the year, the majority of our investigators are going to actually be dosing patients. So this is huge. This is really very exciting. And I couldn't be more pleased to be able to be a part of the program. So that's all I have. Thank you.
So just to break things up just a little bit, because we're going to have a lot of presenters, 2 things I wanted to underscore about what Chuck and Christina presented. They're both from NCATS and doing really exciting stuff. What Christina is presenting, I think, is a really interesting example of crowdsourcing mixed with proprietary incentives. So I don't think we need to have an either-or situation. There's no reason that open source and crowdsourcing can't be mixed with proprietary incentives. And in her case in particular, one thing that I'd like to point out is that new use patents might be really valuable because there's not an off-label problem. So a new use patent could serve, I think, almost as powerfully as an NCE patent in this particular case. And Chuck, you can correct me on that, but I think that that's my view at least.
No, I totally agree, but one other thing that Christine probably can highlight, she should be proud of it, is how people were incentivized by this and the evidence of that. There are 2 points in this project that I was really surprised about. One was, before the awards were announced, is getting Big Pharma on board and to give an opportunity to put a template forward was a huge step, and whether or not they'd actually participate. That was a big struggle. It took several years, as I understand, to develop the program. But what happened was, once I think the first 1 or 2 companies came on board, everybody else just said, okay, let's do it. So that should be a lesson. I'm not sure what it is, but but it's an observation that it raises the hair on the back of my head. Yes, that there's a power there. The other is after the fact, after the program went forward and awards were made and money was distributed and things got running. The other surprise was that now, we mentioned the second iteration, all the academics said, this is great, we want more, and all the pharma said the same thing, this is great, we want more. So I think those two observations speak volumes to the success of this interesting new model.
So we have next on our agenda Craig Wegner, who is with AstraZeneca, one of the leading pharmaceutical companies thinking about rescue and repurposing, and involved with the NCATS program, and as well with also with a program with the Medical Research Council of the UK. So.
I'd like to thank Arti and the organizers as well for the opportunity to participate in this meeting. And I am representing AstraZeneca. I've only been with AstraZeneca, however, for a year, worked with 4 other pharmaceutical companies before AstraZeneca, and throughout my career I've been heavily involved with academic collaborations, many of which have not been ideal and consequently have some views on how to make them better going forward. So in the presentation, I will talk a lot about AstraZeneca's thoughts about drug repositioning, open innovation, and how we think this can advance medical science and bring new drugs to patients. But also what are the key features that make it work, and what are the key hurdles and challenges that we need to get past to make this more of a commonplace within science, within the scientific community, as opposed to the exception. So first of all, rationale for drug repositioning and doing so by partnerships. So first of all, the compounds that have been generated and brought into clinical trials by pharmaceutical companies offer huge value to the scientific community, to understanding medical science, and potentially to bringing new drugs to patients. They offer an opportunity for us to understand unmet medical needs and to explore mechanisms in human biology. Pharmaceutical companies have finite budget, and that finite budget is getting less as more and more patent expirations. You've heard about that, I'm sure. Pharmaceutical R&D has not been terribly productive over the last decade, and as a result, most pharmaceutical companies the theme is focus, focus, focus. And the result is that for these valuable molecules that we're generating and bringing into clinical trials, we are typically focusing on a small number, often just one indication, to place our bet on in terms of advancing them. And the reasons are shown here. It's budget, it's manpower, it's expertise. We aren't spread across multiple disease areas. It's patent life. However, that's not the best for the patients and for the advancement of these molecules. There are potential other opportunities. Most compounds that are on the market, and I say here by most greater than 75%, the reference actually says 90% of molecules that are on the market are approved for indications in addition to or instead of those that they were originally designed for. So we know that there are, and there's many, many examples, and we can talk about some of those if you'd like. So to combat these issues, we and other pharmaceutical companies, but particularly AstraZeneca, has been one of the pioneers and the leaders in the field of reaching out to academic investigators, doing so in collaboration with the NIH NCATS, with the MRC in the UK, and most recently we announced a collaboration with the National Research Program for Biopharmaceuticals in Taiwan to offer our compounds, our molecules, both small molecules and biologics, to academic investigators for proposals for additional indications. We believe that medical breakthroughs, particularly in the understanding of medical science, but also hopefully in new drugs for patients, will result in that. So 2 case studies real quickly. The first was a collaboration that AstraZeneca established with the Medical Research Council, or MRC, in the United Kingdom. The program was called The Mechanisms of Diseases. AstraZeneca put forward 22 discontinued compounds. These were, about half of them had been discontinued after phase 2 in the clinic, so we knew that they covered target and had manageable tolerability in humans, and consequently were ready for application in another patient population. The other half were preclinical tools of exciting new mechanisms where we had strong selective compounds. We received over 100 proposals from 37 different institutions in the UK. Like with the NIH and like with Christine just mentioned, these were spread over multiple disease areas, which is shown in the pie chart in the upper right. The proposals were triaged by a joint review committee in this particular collaboration, and ultimately 15 programs were funded over a 3-year period, with the MRC providing $11 million to fund those. About half of those, 7 of those 15 are for clinical programs, 8 of those are for preclinical only programs. Christine's already mentioned the second case study, which was the NCAT's program discovering new therapeutic uses for existing molecules pilot program. The ultimate goal here, as she mentioned, was a phase 2 validation study for new indications. 8 companies were involved, 58 discontinued compounds. 9 programs were ultimately funded. 3 of those happened to be with AstraZeneca compounds. The real key here in the discussions here was to understand how we make a 3-way collaboration work in both of these cases. And the diagram on the right-hand side tells you how you do that. And basically it is by setting up 3 different 2-way agreements as opposed to a 3-way agreement. So a memorandum of understanding with the funding agency, with the public body, grants between that relationship between the NIH and the investigator, and a collaborative research agreement that covers confidentiality, intellectual property, publication rights, etc., between the pharmaceutical company and the industrial partner. So where do we think we should go next with this? What have we learned from these pilot programs, and what do we think we should do? So I've mentioned the MRC and the NIH. Our oncology colleagues, and this isn't only true in AstraZeneca, but it is well emphasized and we have success with it in AstraZeneca, is the oncology community has been doing this routinely for both preclinical and clinical compounds, where toolboxes of molecules have been presented well prior to registration, made available to major institutions that have cancer focus, as well as the NCI, and multiple investigator-sponsored studies, both preclinically and particularly clinically, have led to new discoveries that have ultimately benefited oncology patients. So we believe that the way forward is to expand upon this, and expand upon this in 3 critical ways. One is to not just do this for early clinical, but also to do this through other stages of discovery. So the identification of new targets, the identification of lead materials by making open our high throughput screening facilities and compound libraries, as well as making additional offerings. We also think that it's important that we go to other parts of the world. There are great scientists in the United Kingdom and here in the U.S., but there are great scientists that exist other places in the world as well. So are there more ideas to be had? One of the interesting things is there was a pretty good overlap between the compounds that we made available to the MRC collaboration and those that we made available to the NIH collaboration. We didn't receive one proposal that was identical between those two, which was interesting. It speaks to the diversity that Christine already emphasized. And the final is expanding the list of compounds that are available, including more biologics, and there's issues with doing that from a pharmaceutical and development point of view, but also including compounds that are live in our portfolio, which have been the chosen jewels of the pharmaceutical industry and sort of off limit even within within companies to drug repositioning. Okay, so what are the cornerstones for success? And it really is that both parties must open up and most parties must benefit. So if we keep those things in mind, that's what needs to happen. So first of all, it has to be an open collaboration. We have to create partnerships that work, and that has to be where both parties are freely able to share their opinions, their questions, their criticisms, and are sharing in the risk-reward of the program. Pharmaceutical company, in this case AstraZeneca, makes the first gesture by opening up a compound and a description of it that we're willing to accept proposals for, or tools such as our high throughput screening tools that I mentioned before. We need to have template agreements or agreements that allow us to interact without each party negotiating for position, and the NIH really helped us out with this. And then finally, it's not about the pharmaceutical company saying, hey, we would like a proposal for you to do this with this compound. It's about really opening it up to concept and new ideas, so that both parties are bringing something novel to the table. The AstraZeneca with the compound, the investigator on the outside with the concept proposal, and that leading to further discussions and ultimately a full proposal and going forward. Not all courtships work. So even the best concept proposals, sometimes as we got to working with the investigator and they found out more about the compound, they felt that it didn't fit. We felt that their ideas weren't going to answer the question related to the disease, et cetera, et cetera. There are many reasons why ultimately we shook hands and walked away and said, this has been a great discussion, but we aren't aligned on where we need to go next, or we are aligned that there isn't a path forward here. Okay. Next, both parties need to be empowered to say no or yes. I just said that. It needs to be resource efficient to us and to the investigator. We leverage some of the things that we already have, the knowledge, the study reports, the data, the compounds, the investigator, also their background, but then the funding from a third party, from a public agency, has really facilitated these. And the fourth that's important is this transparency up front. This is how the program works, and so no surprises down the line. Now, there's two key hurdles that need to be scaled. The first is that the traditions of the past, that this is not about universities gaining more funding from pharmaceutical companies, that it's really about a scientific collaboration. And the second is that there's a really small intersection between those ideas that we need to bring forward. You need to have a patient-ready compound. You need to have an idea from an expert that overlaps that, and it needs to be into an important unmet medical need. And that overlap is exceedingly small. And so the more you can grow each of those, the more likely you are to have a really good idea. So this is my final slide. And just to summarize, I think that we can do a lot together. It's about unlocking the collective power between industry, academia, and the public partnerships for the advancement of medical science and for the benefit of patients. It requires truly open collaboration and a broad outreach. Partnerships have been piloted and shown us the path forward and have been exciting to both parties and look to be successful, but many are still stuck in the traditions of the past. And as has been mentioned by others, the time is right. There are bold steps that are necessary, but there are parties that are ready and willing to make those. Thanks for your attention.
Thanks, Craig. Because we're going to switch gears a little bit from some of the rescue and repurposing / repositioning approaches, I did want to underscore that one piece of the puzzle that still hasn't been solved, and I know that there'll be several people in the audience who will be speaking a little bit more to this later, is how you deal with incentives for the situation where it's an already approved drug, already generic, and you have a new use, but there's no exclusivity associated with the new use because it could be used off-label for that new use. So that's different from Christine's situation where the drug has never been approved, and we'll be talking about that. But now we're moving into something completely different, which is looking at pre-competitive collaboration and what advantages pre-competitive collaboration can yield.
There's no pointer, is there?
There should be.
Oh yeah, maybe here.
Yeah.
It's fun in Washington. People from Toronto feel at home. We have the cracked up mayor comment, and so. So I'm going to talk to you about 10 years of what we've been doing in a collaboration with industry to put science into the public domain. So we are an organization that is funded by the Canadian government, the Wellcome Trust, the Gates Foundation, and 9 pharmaceutical companies, and we never file for patents ever. And that's crystal clear in our ethos, and you can imagine how much headache that avoids. There's more lawyers in this room than I've seen in a decade, because we don't have any. And so what we've done is we're an organization that's a basic science organization, and we work on determining the shapes of human proteins. So human proteins are what drugs act against for the most part. And if you have the shape of it, it's easier to design at least the starting point of a drug. So we've done 15% of the world's output, and we continue to do 15% of the world's output every year from our labs. We've also decided in a project started by GSK and us to start making completely new molecules to using our structural information as a guide to proteins that no one's ever worked on before, with a pre-commitment to put that molecule into the public domain for everyone to use with no strings attached. And this is a really interesting idea where we take advantage of the medicinal chemistry expertise in pharma. That's their core competency. You'd never get access to that before. And they make molecules that they pre-agree to put into the public domain. Just one molecule, but it's a very, very good one. And now 9 pharmaceutical companies are part of that effort. All 9 are contributing internal, they're funding us for one part, and they're also contributing internal chemists to make novel molecules. It takes about 18 months to make one of these molecules, costs probably together about $5 million to make one of the quality that we're putting out into the public domain. But I'll show you later how much value that has to both the scientific discovery and to industry. And so the impact of what we've done, I mean, it's interesting when you start projects, you always talk about process. You heard the last 3 slides because they're just beginning. It's the success is doing the deal. I mean, ultimately, we hope to be showing the examples of when the drug was approved, because that's the only output that matters, right? And so we're fortunate to have done this for 10 years, and we have lots of examples. I've just put 3. This is an example where a biotech company in the Boston area used one of our molecules. So this is the shape that we determined, put into the public domain, as well as a how to use it. And they used it to develop this molecule that's, I think, in late preclinical. Stephen Fry at UNC. This is a molecule for a pediatric cancer. We created the template shape of the human protein, and using that, he designed this molecule. They've started a little biotech company, and they're putting that into preclinical. This is a molecule that Glaxo told us about. GSK said, hey, you know what? There's an interesting class of proteins called bromodomains that nobody worked on, but they did internally. They running into a wall as to which indication. So they told us that, why don't you guys work on it and put some molecules into the public domain, which we did. And it was a very interesting timeline. So Glaxo told us about a Mitsubishi patent where one of the molecules was disclosed and told us, you guys should work on this because this formerly undruggable class of proteins appears druggable. We identified a doctor at Harvard who looked after patients in which the target of this putative drug might affect the cancer. So together with him, his name's Jay Bradner, we had the chemistry plan. We made the molecule called JQ1. JQ1 tells you how much hint we had from Glaxo. You know, when you screen these high throughput compounds of 400,000, none of the drugs is in there, or none of the good molecules in there. You have to do another 18 months of chemistry. We had to do probably about a half a week of chemistry. That's how much the hint the pharma had given us as to what you guys should do in the public domain. But nevertheless, we're using that molecule, and we published a paper in the Cancer and showed it had beneficial effects, a very, very rare cancer. GSK had run an internal program on sepsis that they realized this is not going to be a sepsis drug. This is repositioning in action in the open, FYI. The drugs that are being repositioned, as Christine will tell you, those drugs, the cannabinoid ones that are on the NCATS list were probably made 10 years ago, right? So it's about innovation, but it's how fast these things can happen. It's more important almost. So anyway, so we're talking about in real time, this goes out, and of course we may put this into the public domain, and hundreds of labs took it up. Now it's probably 500, and a doctor from New York and a doctor from Boston, whom I don't even know, took that molecule that we'd given away, did an experiment and said, you know what? That molecule works in multiple myeloma and AML in sort of patient-derived models, not in clinical models, which then caused GSK to resuscitate its program, which had been parked in the lingo of the pharma, and they did their first in man in March, together with the physician we identified. Now, the timelines of this are surreal. This isn't repositioning, right? This is from a patent to this. And you can compare it with Gleevec, which is the home run personalized medicine story, right? This is a fantastic scientific saga from a precursor company to Novartis to the drug. And it took 6 years from the initial disclosure in the patent literature to the first in man. 2 years of that were negotiations between one of the companies and the institution, and negotiations that went south, such that the doctor had to leave that institution to another institution to sign the exact same deal that the first institution didn't sign, and then it made it to patients. 2 years, and these are a great drug, of people dying because no one could come to a deal. That's sort of surreal, right? Well, it happens, and that's why I guess part of the reason we're here today. But I know you couldn't prescribe this, and it may never happen again, but it did happen, right? And in large part, it's because the willingness of the companies to disclose information, the willingness of our scientists to give things away, the willingness of Oxford and Toronto universities not to get in the way of our no IP policy and accept those terms, and then the willingness of other professors to publish, but they get good papers, so that's not hard. So I guess one other question is, why? Why in the world would we get $70 million over 4 years from pharma to make new molecules and give them away? I mean, that home run story is one of them, but here's an interesting thing on, in quotation marks, how innovative we are. You know, what industry wants now is they're going to downsize internal R&D and access, in quotation marks, innovation from academia. Well, they're SOL, because the interesting thing about the human biology is that in the only scientific discipline in the world where it's bounded, there are 20,000 genes, and there are only 20,000 genes. Come back in 10 years, there's not going to be new genes, right? Unlike stars or chemistry or this, where the science is infinite. So you can box, and you can start charting research activity in areas. So you can go into the digital libraries and ask how many papers are there on gene 1, gene 2, gene 3, and when you do that, for example, in this protein family, which is an important one for drug discovery, this is the number of papers where the name of the gene is in the title or the abstract, and these are the human proteins. Most of the research activity on the planet is clustered on a small fraction of the genes, and believe it or not, most of these genes were hot in the mid-90s, right? So where are the repurposed drugs coming from? All of these proteins are in here. So we keep looking in the same little area, and there may be opportunities, but they are pale in comparison to the opportunities out here. Now, one might argue these are important genes. They are. But these little colored stripes are when people do different kinds of experiments, and every time there's a bar, it says, hey, that one's pretty cool, right? But it's impossible to work in it. If you live in the peer review world that we work in, you come into a grant and you say, I want to work on protein number 351. It's really cool. I know nothing about it. I want to find out about it. There's no elegant hypothesis. There's no people who cite your work. There's no track record on it. It doesn't even get reviewed, right? By contrast, if you write a grant on gene 20 where there's lots of papers, you can prove how smart you are by connecting dots and apparently being really brilliant, and you will get funded, right? If you want to go, think professors don't work for money, we work for peer recognition. How do you get peer recognition when there's no peers? Right? You get a lot of peer recognition. You want to get invited to talk in Barcelona, work on number 20. Because it's like the Facebook of science, right? There's a little community, and they love each other, and they go to meetings, and they, oh, isn't it nice to go to the meeting every year, etc., and they do this. But this is, I mean, it's a little facetious, but the data aren't facetious. This is real stuff, right? And there is an underlying reason, and it's a lot because of what I speak about. And so if you access innovation, clearly the academic system is not structured well, or the funding system is not structured well to do it. It has important effects on industry. Because if you look at the, remember, most of these were hot in the 90s. Where are the patenting? The last time I did it was 2009. The same place. Because the key opinion leader from Harvard with the bow tie goes into the drug companies, where his buddy is the head of research or whatever, talks about the exciting new work he's doing where the mouse is doing all this interesting stuff, right? There's lots of papers on it. These are data-driven organizations. They follow the literature. If we provide literature in this, under this light, they will do research under this light. You wouldn't believe how hard it would be if Craig went to the VP of Oncology and asked for money to work on 351. It's a $20 million bet, no papers. It's a pretty hard experiment. No key opinion leaders, no knowledge of what it does. The answer will be no, right? Where are you going to get the money? To work on something where there's a robust literature, where's a robust literature where we've worked on since the 90s. Okay? So neither academia nor industry is truly structured to innovate, right? And innovate means go into the unknown. Academically, this is not considered innovation because it's using the same tools we studied these here. That won't get you a research grant because that's not an innovative idea, it's not an innovative trick, it's not a cool little micro this or nano that. It's just moving into a new area. You cannot get funding for it. So we created the organization to explicitly work on those proteins and to share the risk, because we didn't have a lot of funding from either. No sector would do it alone. So we figure, you know, the game is get everyone a little to pay, and that worked. We run at 2 universities, but we really have a milestone-driven project, et cetera. Every quarter we have a board meeting, did you meet your milestones, et cetera. And the academics that we've recruited buy into that because we publish 2 papers a week and all in, a lot of them in those journals that everyone likes to publish in. And of course we never patent because that allows us to give away. In 2013, we've given away 1,600 small molecule aliquots to people. No MTA. Can you imagine how much legal time if you had to give away 1,600 in 1 year? And these are new molecules. We have a click wrap MTA, and the click wrap MTA, you just click, you don't have to get anyone to sign. I agree not to eat it, I agree not to sell it, I agree not to give it to people, and I promise as hard as I can to put it into the public domain. And that's it. And companies now, Janssen is giving us molecules, and it says, could you guys please give me my molecules away under your thing? Because hell, if I have to go through my lawyers to give it away, and so they do one thing with us, and then we communicate it to the world. But this is in the public interest, right? Because these are molecules now that are getting out there uninhibited and allowing people to work in the nether areas, as it were. And as an objective quantification of sort of the past decade, these are our research outputs where we worked on and solved structures, right? And this is continually, this is from 2012, papers, it's that same set of proteins that I told you about. These are 2012 numbers, right? There is one, BRAF, that's an exciting melanoma target. It aggregates, half the world works on it now. We don't need half the world working on one thing, remember? The other interesting and terrifying thing is if you plot Canada publications on this, it's exactly the same shape. And that's perfectly explicable because funding is parochial. Look, we're here all United States talking about US. If I'm in Canada, we're talking about the same thing. The scientists do the same thing. They all read the same literature. Nobody's smarter than anyone else. Everyone's got the, I know what to do next. But I write to the Canadian government, I get my grant. My colleagues in the US write to the US government, they get their grant. It's exactly the same science. And then Germany and Spain and Australia. And as a result, you get clustering and superimposable patterns. It's bizarre, right? Like this protein in Canada will attract, it'll be a little peak. If you go to the UK, it'll be a little peak. It's kind of distressing, yet pitiful use of global resource. Another example of this model is about why it works. We're not really we get a lot of funding. We're not really incentivized to publish. So the Economist published this, a lot of academic experiments can't be repeated. But what Rand did a study on us, and they said this kind of model, where industry is part of it and industry values reproducible science, we made sure we did reproducible science. And one company joined us, and I said they rejoined us after they refunded us. Why'd you refund it? Oh, we could just repeat your stuff. I thought because you guys are so smart, because you're so fun. No, we can repeat your stuff. That's the bar, right, for industry. And it's not, of course, all the story, but it's an important one. If you file no patents and you explicitly do it for science, you can collaborate with anybody around the world. It takes approximately 3 microseconds to do the deal. And so what next? So what next? We've identified 3 hospitals in Canada where the director is willing to engage in collaborations with the patients, all preclinical so far, all in the public domain, accessing patient tissue, accessing the clinician's brains, and agreement up front, no matter what we file, will go into the public domain. You can imagine the incentive for the patient now, because we get pharmaceutical expertise. They're really good at what they do. But an explicit, this is a never patent project. It's off the table. There's no conflicts of interests, right? And we're the sort of trusted intermediary to make that happen. But now we've got 5,000 Parkinson's patients who are going to be involved in studies with these new molecules, right? And it's all about the science. And so nobody is going to start to feel, oh, should we do this? And of course, in Canada, it's a lot easier than this country because there's a lot more fuss about that down here. And so what I like to end in is that, why don't we let you guys play on the end where everyone else is playing, right? I think there's an enormous opportunity. If we took, Canada has 1% of the global R&D budget. I think it's a tiny fraction. But $1 spent on this side is better being than $100 spent on an old chestnut over on this side. Now, I will not win this battle, FYI, because the Canadian peer system is just like the NIH peer system, etc. But it's my dream, right? Thank you.
So we have Pearl Huang next. I'm going to have a provocative question that I'd like all the panelists to think about. In your repurposing and rescue projects, how much of have you gotten into the tail end of what Aled was talking about? Have you gotten proposals that address the nether regions of the target world? So think about that as we hear more speakers. Pearl is from GSK, and I'm actually really curious about why GSK did that open access project. So if you could speak to that, that would be great,
Sure.
too.
Do I control it this way?
Let me
Oh, okay.
do it, sorry.
All right.
I
So,
have to play all the jack of all trades here, so.
okay, terrific. And I'm a drug hunter, so I've never, like Aled, been in a room with so many lawyers or articulate people before. So I think the reason we're all here is because there's a problem. And as I was listening this morning, you know, drug discovery and development is something that takes a lot of time, maturity, and then what you're trying to do is create value for the patient, right? There's unmet medical need. So in the beginning, when it's just early ideas, you know, the value gets up to a certain peak, and then as it matures, as we were talking about in the earlier session, you start to get more ideas, but they have less value. The problem is that everybody thinks every part of their contribution to this process is the same. And that's where we get into trouble. I live in this space right here. I'm in the early discovery and development space. And so what we've decided collectively as an organization, but also I think as industry, because there are others here of like mind, is that there's a lot of risk in this space. It's really hard to make a new medicine. So in order to do that, we have to collaborate, we have to partner, and we have to share the risk. And the other side of that is, of course, we have to share the reward. So I'm going to describe a couple of the projects and of the activities that are ongoing at GSK. I'm going to focus mostly on the area where I've been working, and I'll mention at the end another theme on the variation of these partnerships. Today we just announced the creation of a new biotech together with a VC company, and it's operating on the same kinds of principles. There is an open innovation aspect to what we do, but the reality is we are a for-profit business. And in fact, I would argue that the highest quality medicines, and I think Jay Bradner would also argue this, actually comes from the private sector because people are incentivized to make the very best molecule to take forward. And that might be at this stage of the invention where it's not so interesting to an academic, but the scientists within a place like GSK are willing to push it to that extra effort. To incentivize those people and to incentivize certainly many of the academics that we work with, you have to appeal through their greed. And it's greed not just for money, but it's greed for glory, and it's greed for access to more ideas, more funding, and the opportunity to solve problems. So I'm giving you my email address if you have any questions. Next slide. Or do I do it?
Oh, you can do it.
I can do it?
You can
Okay.
do it. You should have a clicker with you.
Oh, okay, great.
Yeah.
Okay. So I used a couple of company slides here. GSK is a big global pharma company. We invest over £3.5 billion a year in R&D. That's a big pot, right? But our success rate is no better than anybody else's. As I said, making a medicine is very difficult. And so a couple of years ago, actually, GSK decided for two reasons. One was to expand our ability to interact with other people in the drug discovery world, but also what we know, and it's still true, is that most of the world doesn't like us very much, right? They think we're secretive, we don't share, you know, we don't publish, or we only publish after something is either of no value or of great value, but nothing in between. And that squashes everybody's ability to learn. So this is from our website, trustandscience.gsk.com, and it reflects really the commitment of the organization to open up our science, and you saw an example of that in the last talk. My colleagues at GSK have been doing this for a while, so I've been running the DPAC program for about 2 years. But several years ago, our Diseases of the Developing World, which is located in Tres Cantos, opened up an open lab to deal with the diseases of malaria, tuberculosis, and leishmaniasis. And so this is a very unique situation where they invite academics from all over the world who work in these disease areas to come and work with them in Tres Cantos, to have access to our chemical collection, to our technologies, our high-throughput screening. And for the time that these academics are inside of GSK, everything they do is shared with all the other academics as well as the scientists at GSK. And then they can take everything they've learned with them when they go back to their home laboratories. And so that's been a very successful example of one way that open innovation is working inside of our company. Bill Zurcher in North Carolina at the RTP site is well known for his success in building a kinome test set and distributing it throughout the world. And so what this is a test set of kinase inhibitors. As all of you know, the human genome was sequenced and the kinome's been sequenced, and so we now know the identity of every kinase in the human body. And so we're pretty good at making inhibitors of the different kinases with some selectivity. We're getting better and better at it every day. And so there are a lot of published compounds, and then some that weren't quite published but that were complementary. So what Bill did is he put together a collection of almost 400 of these individual compounds that were all really well characterized, so they were known entities, and he offered them to the world. He said, take the set, test it against your biology, test it against your target, see what you can learn. This is still in the early discovery space, so we're still talking about this area here, and I think the reason Bill was able to convince the organization to do open innovation in this space is it's still quite early, right? There's a lot of invention that has to occur after you make this observation. What's interesting is I just talked to Bill about this the other day, and I said, what has been the biggest challenge in getting this out to the world? So I think there are over 140 labs that have taken this on. And he said, actually, the academic institutions don't like the idea that we don't want anyone to patent anything that comes out of this, right? So the deal is, if you take this set, what you promise to do is to publish your results, if you can. And there are a lot of places that don't want to do that. They're academic, nonprofit institutions, and they want the right to file a patent and then sell that information to someone through a license. And I have to say, as DPAC moves around the world and negotiates agreements, we see that as well. So I think there is a shift of where, of who wants what in this space. Just as an example, in terms of how this test set has generated value, I say, I ask Bill, how many new projects have come into GSK for this? The answer is, at this point, no. How many new targets have been validated to the point where we're convinced they're worth working on? The answer still is no. But what we have been able to do is improve the ecosystem, and so people who have had access to this data set and who also have been able to publish their results are able to attract more funding so that they can take it to the next level. Okay. So I'm going to talk a little bit more about DPAC. I talked about how we're trying to collaborate and also about the structure of these collaborations, how we're sharing risk and reward. The idea here is to take the best of what we are inside of GSK R&D and combine it with the best of the academic world. And so what do the academics have? They have deep knowledge of targets, biology, and disease. And what do we have? We have technologies, we have drug hunters, we have medicinal chemists that I would argue are some of the best in the world, and these are the people who can help us meet unmet medical need. So we scour the world looking for people who want to collaborate with us. It's a very simple premise. Come in and work with us, be exclusive partners with us for the generation of a new medicine, and go with us as far as you can go, all the way to candidate selection in the case of the initial contract. But we also ask these individuals to be part of the advisory clinical development team. And the payoff for them, in addition to all of the funding that goes on to enable the work that needs to be done, is that they get to share a royalty share in the final product. We look for academic researchers who want to be involved. Obviously, we're asking for a deep commitment from them and bring something unique to the collaboration and also have really unique insight onto how to solve a problem. Many of the diseases that do not yet have therapies have been around and well described for some time. But we recognize that there are many people out there who have unique insight, who haven't, for one reason or another, maybe it's granting, maybe it's because they work on an obscure protein, haven't been able to get the resources to take their ideas forward. In terms of managing the relationship, these projects are jointly run by a DPEC scientist and the PI, and after the contract is signed, and I have to say that in North America, we have not been able to sign a contract in less than 11 months. Whereas in Europe, we get them done in 4 to 6 months. So all the lawyers in the room, pay attention to that, please. So once that contract is done, those two individuals have full decision rights on running this program. So there are no committees, there are no reviews, they don't have to ask for permission, they've got a budget, they've got a plan. They have a steering committee to advise them, but the two of them have the decision rights, and we think that's really important. What we find is with these projects is that they become very intense. And so people are talking to each other daily. The labs are meeting sometimes weekly. We have scientists from the academic institution coming into GSK, scientists from GSK going into the academic institutions. Because of this contract, that's all covered, and they have the freedom to do that. And I should also say, even though it takes us forever to get these contracts signed, one of their features is an equitable approach to IP. And so we work in this early space. Most of what we do will fail. That's the reality. You can do the very best science and the very best drug discovery that time will allow you to do, and you'll still fail. So something that means was very important to the academic scientists, but probably even more, if not just as important to the scientists inside of GSK, is that whatever work is done under this partnership doesn't get put on a shelf. So what happens is if GSK at any point decides to terminate the project, then the IP goes back to the academic institution, and the academic PI has the freedom to go and partner with someone else, raise funding, and put it into any structure that they desire. Okay. So I have 5 minutes, and so I'm just going to breeze through this. This is the structure, basically, of what our interactions look like. You can see these are the chevrons of traditional drug discovery and development. Our programs are focused right on the second half of this. And what we do is we fund the academic activity in the laboratories. You see that on the bottom with the dollar signs. And then GSK provides in-kind resources to complement that, drug discovery, applied science resources. And we would argue that the highest quality resources still resides in private industry. We have established 9 of these partnerships. They're ongoing right now. This program started in the UK, so our first 6 are in the UK and in Europe. But we were able to get some in North America, 1 in Canada, 2 in the US, and we're actively pursuing in this geography. Now, I mentioned that it takes us a long time to get these contracts signed. We have several under negotiation right now. But I should also mention that when we scour the world and look for these opportunities, we have to keep a record of everybody that we talk to. And so we have seen to date about 1,200 individual opportunities. You see that we've started 9 programs with a couple more coming along. So our number is about 1 in 100, right? That's because what we're looking for is something that's not just quality science, but also something that's ripe. It's ready for the application of all of those resources. So as somebody who has to run this team, my question to my people was, why are we spending 99% of our time listening and learning about things that we're never going to be able to prosecute? So what we decided to do was to bring opportunities into our organizations to run a contest. And we called this the Discovery Fast Track competition. The idea being that people would come to us with early ideas for a therapy, and the reward, the winners of the competition, would get access to all GSK resources so they could take it to the next step. We opened this up to everyone in the US and Canada, in part because competition law was pretty complicated for us to do this worldwide, so we thought as a pilot we would just see if we could get this to work in this geography where we were most familiar. And again, these are the resources that would be made available to the winners. We opened this contest on the web. We had to build some tools, we had to build some communication, and what we've learned most of all is that we had to educate. We had to educate people about what drug discovery was, and what and how it was done. We opened this competition on May 19th of this year, and on May 24th, we got a cease and desist order from a lawyer at the University of San Diego who said that we were trying to steal ideas from academic institutions. So that wasn't the case. If you went to our website, you could see that we had everything in place. We had the MTA posted, we had the CDA posted, and the only grounds for disqualification for anyone entering this contest was not getting a signature from their tech transfer office. We managed to meet with the head of central research, head of research for the University of California system, who after several conversations said, this is perfectly fine, everyone's allowed to do it, go at it. And so we had, partly because of that controversy, over 10,000 hits to our website. We had over 400 people opt in to say that they would consider applying. But the contest was open 60 days, and we got 142 applications. And I think there was another 160 day in there somewhere where you got about 160 applications, so maybe that's the determinant. But this is just a map to show you in the United States who applied. The people, the laboratories and the PIs who won are starred there in red geographically. And I have to note that we got applications from 4 Canadian provinces, and one of our winners is in Quebec, in Sherbrooke. And we would have never known to go up there. It's an outstanding proposal, and we're very excited. In terms of proposals by therapeutic area, we had a pretty good distribution. And my guess is if you superimpose this on what the NIH funds, you'll see the same kind of thing, right? A lot of antimicrobials, a lot of oncology, and then a smaller number of proposals in other disease areas. And here are the winners. These are scientists. The way this contest was run, I have to stop. So I'll go, give me 3 minutes, okay. So the way this contest was run is we used the web, and so people submitted their ideas non-confidentially. We had a blinded panel of scientists within GSK who reviewed those proposals. They blinded, meaning they couldn't see what anybody else was thinking. We thought that was really important. We brought in scientists from very different backgrounds, all of them with a good track record in drug discovery. And then the good ones kind of automatically came to the top. We then brought in a subset of the finalists. We determined there were finalists, and we brought them in, and we had face-to-face meetings with them over 4 days. And from that, we chose 8 winners. 4 of these winners are in the antimicrobials or anti-infectious area, and that's partly, it's definitely because their proposals were so strong, but also because in terms of target validation, infection, I think, is a place where it's very clear. However, we're also working in other areas. We brought in some new areas of disease to GSK through this. Here's our fellow from Sherbrooke in iron overload disease. We're looking at male fertility and cancer. Not everybody who entered this contest and won, as you can see here, wanted us to publicize who they were. They would prefer to work in the shadow of anonymity, and we grant them that because this is still very early research. And what I tell people is there's a greater chance you'll fail than succeed. Okay, so this is DPAC, who we are. This is a small group of people who are, all of whom have a track record of success in drug discovery. We're all drug hunters. We have a combination of biologists and chemists. The question marks are we are hiring, so we're looking for people to join us. The other thing I wanted to point out is it's structured, we're flat. Almost all of these people report to me, but the people who are outlined in orange are actually on secondment. So these are people who come in and say, this looks interesting. I want to see if I can run a project through this mechanism. And so they come in for a year, maybe 2, and then they take their project with them and go elsewhere inside of GSK to make it happen. There are a lot of pockets of innovation and expertise within GSK, and we want to make sure that each one of these projects goes to the place that they fit the best. So that's it for me. Questions?
So we have 2 more speakers, Bernard Munoz and Dominique Pahud from Kaufman. Bernard, I think you should go next because Dominique, you wanted to go last, right? Sure. Okay. Bernard, let's see if we can wrap things up in the next 25 minutes or so we'll have plenty of time to respond to one another, because I know that Aled in particular, for me, raised some very provocative questions.
How do you get me to, how much time do I have?
So why don't you have about 12 and a half, would that work?
Until 12 and a half?
Yeah, would that work, or is that going to be too low?
Well, I've got 11:30.
Oh, 11 is perfect.
No, I've got 11:30 now on my
Clock, right.
clock.
Yeah, that's right.
So until when?
11:45.
11:45, okay. Where's the clicker? Where do I get my stuff up?
Just 1 second.
Oh, okay. Good morning. Lots of interesting discussion this morning. I will come back to some of what you've heard, trying to maybe draw some general conclusion, and also add to the information, the model that have been described to you, and maybe suggest that there's even more out there that could be used to do innovative drug discovery. So, this is a slide that I won't go into detail because you hear about it all the time. I'd rather save the time for something more productive. I just want to draw your attention on a chart at the bottom left, and it shows the the amount of money that is being spent for each drug that is brought to market by various companies, some big companies and small companies, and you can tell that those amounts are, especially at the top end of the industry, astronomical. Now, you oftentimes hear that it takes about $1 billion to take a drug to market. This is a bogus number, and even if it weren't bogus, it would be irrelevant. What really matters for society is how much R&D money a company spends for each drug that they bring to market. And depending upon who you're talking about, that amount varies from $2 or $3 billion to upwards of $10 billion. It is clearly unaffordable, and it raises all kinds of problems that you can imagine. I won't get into that. So the model is broken, and we need to fix it. You've heard some approaches earlier today that look very promising on how to fix it, and I'd like to add to that. We heard about drug rescue, and clearly that works. Now, it works so well, in fact, that it begs the question, why aren't we doing more of it? The proof of concept has been successful, and yet I don't see the industry rushing out there with, there's thousands of drugs that could be rescued in their vaults, and no one is rushing with, to the scientific community and say, here it is, you've done it on 38 molecules, do it on 2,000 molecules, and everybody will be better off. In fact, there is, you could imagine some kind of an exchange where the industry would contribute those molecules that really do no good in their vault, and have some standard agreements, and the scientists of the world could go and look it up on the internet and decide what it is that they're interested and under what terms they could access those molecules, and it would be very simple, very efficient, very cheap, and yet it is not happening. So there's something to ponder there. One other thing that I want to mention about drug rescue is that a small company that I work with, when the 38 molecules were made public, actually posted that list of 38 compounds on its website and invited the scientists of the world to submit proposal that would repurpose those drugs for something worthwhile. And there was no assurance when that was done that anyone would care, that anyone would even bother to visit a website, let alone spend quality time finding out what they could be used for. And in fact, this is just exactly what happened. And it boggled my mind when my friend called me on the phone and said, you wouldn't believe it, pharmacologists coming out of the woodworks and spending their a lot of quality time with their constructing very elaborate arguments why this particular drug might be useful for this or for that. And when he approached some selected pharmaceutical companies after the experiment saying, hey, NIH is not going to pursue this, we really would be interested in spending our money on this thing, could we have access to it? The answer, we're really too busy to worry about that. So it's unfortunate. Drug repurposing, we heard about that. The lesson there is that what we do in, let's say I worked for the pharmaceutical industry for 30 years. What pharmaceutical company like to do is focus on stuff. This is what business people do. We focus. We're very good at focusing. But everything we heard this morning is that innovation is not a result of focus, it's a result of divergence. You got to expand the perimeter of what you're exploring if you're going to discover new application, new drugs, new molecule, and this is not what we're doing. We keep, researching under the lamppost. Clearly, we can do better there. Public-private partnership, we talked about it. I won't go into that. It works, and it works in not only boosting innovation, but also in doing it very, very efficiently. I think Al mentioned that. You can look at what organizations such as Medicine for Malaria Venture are doing, Gavi. They got a few dozen people, less than 50 people, and a budget that is less than $50 million. And yet, what they can achieve with such limited resource base is nothing short of incredible. They basically bring a drug to market for less than $100 million when it costs big pharmaceutical companies in the billions to do that. But there is other very interesting, very encouraging models that I'm going to talk to you about very quickly. One is a micropharma model. Now, micropharmas is a new species, literally, that appeared on the market about 6-7 years ago, around the year 2005. And those are skeleton companies, typically less than half a dozen employees, or less than a dozen. And you might wonder, what can 6 people do when, company XYZ, Sanofi, has over 10,000, GSK over 15,000 scientists out there? Well, the answer is they can do actually quite a bit, and they can do it on a shoestring. I have tracked the population of those micropharmas, and there is here it is. There's about 35 companies that are available, and you may recognize some of those names. Now, there is several things that is fascinating about that population is that we know that the drug R&D business is high risk, that the odds of failings are incredible. So you would think that, very small companies with very limited resources trying their hand at it, after 6 or 7 years, most of those 30 companies would have failed. 29 out of 30 would have disappeared, and the 30th would probably be very sick. As it turns out, none of those companies have failed. Now, what causes them to defy the odds? It's not that they have a lot of money. It's not that they have a lot of people, but they're still succeeding. Now, one thing to ponder. The other thing is that they not only fail, but collectively, 22 of them have gone out and raised money, and they've raised over $800 million from people who don't give it away very easily, as we know. So what causes very smart people to give so much money to those companies? Apart from the fact that they beat the odds, it's something to ponder also. Perhaps the answer is that 6 of those 22 companies that have raised money have actually gone through exits, as they say in the VC business, and created over a couple of $2 billion of shareholder values for their investors. So clearly, you can imagine, a system of very passionate people. Most of those companies were founded by refugees from big pharmas. Refugees people who had really big ideas, really oddball ideas, too big and too bold for their former employers. So they go out there and create a company and to pursue that idea. And failure is not an option. And they change the odds. So enough for small pharma. Let me talk to you about the data capture, observational data flow capture. Now, the way we do, one reason why research is so expensive in this country is that, drug R&D is basically a data collection exercise. And the randomized controlled trial that we've been doing since the 50s and the 60s is a very rigorous way to do science. There's no question about it. It is a gold standard, but it is also a very expensive way to collect data, and the amount and the quality of the data that it collects is amount of data is limited and the quality is somewhat uneven. There's a lot of things that has happened in the realm of science and technology since randomized controlled trial, emerged back in the 50s and the 60s. Today, you've got technology, the biosensors, the apps, that allow you to collect enormous amount of data per individual, per every minute, every hour. I mean, I wear a Fitbit, and it literally tells me at the end of the day everything that I've done in terms of my effort and how many steps and how many stairs and when I did them in a day and all that sort of thing. And you look at the field, there is all kinds of technology that is becoming available that makes the data capture very cheap automatically. I don't even have to worry about it. I wear it and it goes to some place in a cloud where it accumulates, and it can accumulate for years. It can be just electronic data, it can be pictures, it can be all kinds of things. And the fascinating thing is that, let's say that I get sick 5 years from now, some strange disease. The physician can access that data. I can play the movie in reverse and find out, where I went off track. You can actually imagine an environment where that data flow feeds into something like the IBM Watson system, where it automatically scans it for patterns and to identify disease that are in the process of developing. One of the weaknesses of medicine is that we have no natural history of the pre-symptomatic phase of a disease. Medicine starts when I get sick, but maybe that disease has been brewing for 10 years and I didn't know it. If with biosensors we generate the sort of data that can create this natural, pre-symptomatic natural history of the disease, we have the way to change medicine. We have the way to change biomedical research. And it doesn't cost a lot of money because those biosensors are cheap. You carry them in your cell phone. You carry them in your Fitbits. So something to think about it. Crowdsourcing, that there's an enormous space there, all kinds of model. You may have heard about the open source drug discovery model in India that started 5 years ago, 2008. 6,000 scientists have signed up to contribute their expertise for free in coming up with new treatment for tuberculosis. That effort runs on $2 million a year. So how do you do, cutting edge science? And by the way, it generates about 65% of the papers on tuberculosis. So how do you do, you get 6,000 scientists to work for you for $2,000. It's a pretty powerful model. I talked about my friend at TLS early on, the rare disease platform. You see a lot of innovation there in terms of doing drug on a shoestring. Rare diseases, parents who have the misfortune of having a kid, for them failing is not an option. And they're not scientists, they don't have any money, but before you know it, they are out there raising money and funding the tools to do the research. They're doing the patient cohort, they're doing genotyping, the tissue banking, they're doing, they're funding a mouse model, and then they put it out there and says, come and work on my disease. All the tools are there. I've got high quality data. I've got, everything that you need. You don't need to do it yourself, and that model works. And we'll hear about that this afternoon. So we've got lots of research models that are being experimented with out there, and I will stop there. I could go on for quite a bit, but that's it. Well, there is that's my last slide. There is some issues that I think we need to address collectively if the promise of open source drug R&D is going to be delivered. One of them is that sharing is a great idea, but it takes an infrastructure, and it's not a trivial infrastructure. If you're talking about sharing patient-level data, if you're talking about share anything, you need computers, you need servers, and you need to share it, not only today or next year, but 15 years from now. And who's going to own that infrastructure, and who's going to maintain it? That needs to be addressed. The privacy safeguard for patients. I can contribute my data, but who's going? If I can do some good, but who's going to access to it, and under what conditions? Who owns that data? Who owns the IP that will be created out of the data? All those are important issues that are that could stall the progress of science in and open source drug R&D that need to be addressed. They're starting to be addressed. I think the IOM has run some initiatives on that. So just a quick overview. Thank you very much for your attention.
Last and certainly not least, we have Dominique, who I take you have no slides, so you're going to talk. Oh, you can go up. Yeah, the rest of the panel can come up. Yeah, let me just do a shout out for the Institute of Medicine. I'm on a committee on responsible sharing of clinical trial data, which we hope will address all of those privacy and IP issues and finally pool all that data that's been locked up in RCT trials in companies' vaults.
Hi, everyone. It's my pleasure to be here and support this event. I think I'm going to keep it relatively short so we have opportunity for Q&A. I just want to give you a little bit of voice, give you a better sense of what are some things we do, what we care about, and how we do some of the work we do. So the Kaufman Foundation is the largest private foundation that does work in entrepreneurship. We also focus a lot of our time in innovation and education. My work tends to focus in the healthcare life sciences because of my background. I used to work for many years in the biotech industry. I'm going to sort of describe to you a framework of how I think about our work, and then I'll give you some specific examples of how what kinds of work fall where. So our work falls under probably 3 or 4 different buckets. One is awareness. So what is the issue? Why is there an issue? What should it be solved? How to tackle it. The second thing we do oftentimes is then once we understand there's an issue and start understanding some of the problems, we like to convene a very diverse set of stakeholders to brainstorm solutions around these problems. We also like to then develop tools and perhaps focus on specific areas that we think we can have an impact. And then we also identify people in the community that are doing some of the things we think are worthwhile, and we support them through funding. So let's talk about awareness and sort of distill it down to why repurposing and how we end up in this space. So we're obviously the innovation model in drug discovery is not efficient. It's broken, and it's a lot of things are happening that really should not be happening. And so one way we sort of chose to focus on is the area of rare diseases. These are really highly motivated patients that don't have any solutions, don't have any therapies available. And the resources available to them are not sufficient. And so what we did to bring awareness around the issue is we actually developed a documentary called Hear Us Now. If you want, I recommend you go check it out online. It's hear-us-now.com. And this is the story of the Hempel family and their daughters who have a rare disease called Niebylski. And so really, I feel like it's shameful what happened, what they had to go through, and we really want to bring awareness around these things, and a call of action. Let's do something about this. So as we think about the continuum of, I call it the healthcare continuum. I'm going to repurpose one of the charts that was used earlier today. So if we think about drug discovery in the early stage, some of the preclinical work, first-in-man studies, large randomized clinical trials, and then a regulatory review, and then actual delivery and payment of care. So these are all different dimensions in which innovation can happen, and in which we could potentially try to compress some of those timelines and remove some of the risk. So I'll describe some of the things that we supported to help answer some of those questions. So in the early phase, in the discovery phase, we've really been big proponents of making drug discovery pre-competitive. So we've actually supported Sage Bionetworks, Stephen Friend's work, where a lot of data has been gathered from multiple labs across the world, and they're able to together share data and really accelerate the understanding of the biology of many different types of diseases. In terms of translational medicine, we've also funded IAMI, which is the Institute of Advancing Medical Innovation in Kansas City. And I'm mentioning this example because it's a perfect, a really unique, remarkable thing came out of this initiative. Basically, Oronophen was a product that was described earlier today. It was a collaboration with the University of Kansas Medical Center, the Leukemia and Lymphoma Society, and NCATS. And in less than a year, actually in 11 months, it went from test tube into man, and which is, as you know, really remarkable. And part of it is, yes, it was a repurposed drug, but also was the collaborative effort of these people coming together to try and solve this. As we start thinking about the next phase, right, in 11 months to get a promising clinical candidate with efficacy in humans, how do we get it to market? And so one can argue, NIH, government agencies could fund the remaining clinical trials needed, or how do we create the right incentives? And so we brought together a really diverse set of stakeholders in Washington about a couple of months ago. So these were generic manufacturers, biotech companies, private payers, public payers, wholesalers, specialty pharmacies, benefit PBMs, really are diverse stakeholders of the healthcare system to really brainstorm how do we create incentives for repurposed products to have a role and actually get to market. And so we have actually identified a solution that we're going to be publishing soon. And really, to really shortly describe it, it's a differential pricing and reimbursement tied to the diagnosis. So the product, the repurposed drug, and the new use would have a new market exclusivity terms. And there's a lot of detail that are important tied to how you do that from an operational perspective. The devil's in the details, as most of you know. And so we'll be describing some of those details pretty soon. And then so I guess the big thing we also do is actually create awareness around these solutions. So we develop tools. One of the big hurdles that we see in the context of health, but also entrepreneurship, is collaboration. So oftentimes when people fall in love and decide to work together, a lot of friction happens right away. Once the tech transfer office gets involved, once the lawyers gets involved, and there's let 10 months, 12 months, a year, 2 years happen, and especially for an entrepreneur, they can't afford that kind of time. So we have been very active in developing what we call a health collaboration playbook to describe what are some of the legal structures you may consider, how do you deal with intellectual property rights, how do you deal with data standards, what are the right data standards, how do you share who owns the data, how does the data flow. And we're actually also going to be releasing that pretty shortly and introducing in the community to hopefully remove, again, some friction that happens, unnecessary friction really in the community that prevents more innovations from getting into market. So I think I'm going to stop here. I'm happy to one-on-one later talk to you in more detail about any of these programs, but I'll head back to the panel.
Well, this is a truly remarkable, stellar cast of characters, and I only have the one question that I seeded earlier, which is, I suppose with one preliminary, so is Alad's concern about the nether regions, should we all be as concerned as he is, and are any of your initiatives getting to those nether regions?
So I think it is concerning, actually, and I'd heard Al speak, I think, many years ago. He spoke to the Molecular Libraries Group at the NIH, and I think it really is problematic that we just continue to study the same things over and over and over again, and we know more and more just about just a little tiny bit of the entire landscape. And so actually the program that I talked about today doesn't really address that, but the NIH I think has recognized this now, and we are piloting another program, which is the Eliminating the Druggable Genome program, which is specifically to sort of get at this thing of basically if you come to us with an idea, if you come to us with an application for something to study that we already know something about, we don't want it. We want the one that, like Al said, that it's impossible to get it funded because it's like, all I know is this gene exists and that's it. Can I have some money? And so we are trying to do that. I will say though that for the program that I talked about, we had asked all the companies based on the applications that we had sent to you. And they, again, are only seeing a subset of the pre-applications of the ideas. From those that we sent to you, were you seeing new ideas for uses? And in almost every case, they were seeing at least one new thing that they hadn't thought of, that they were unaware of. And so there's some strength in there. I think those are all in that in that left-hand side of that chart still, but nonetheless coming up with sort of new uses for them. So
the same targets, but still new uses. Yeah, yeah. Good. Anyone else want to respond to that question?
So I'll be a little provocative in stating that, first of all, I agree completely with what Al had said, in that funding and focus and anytime you get into a peer review process, which happens in industry as well as in public funding, you're going to be focused on the existing data, and it's kind of like the person getting their first job. Well, come back to me when you have experience. Well, I can't get experience unless you give me a job, sort of thing. But I think we need to separate innovation from novelty. Novelty is doing something that has never been done before. Innovation isn't necessarily that. It can be putting together several dots and creating something of value that is new, that isn't out there at the present time. And to just echo Christine's comment, drug repositioning is something that has shown innovation because it's shown to move something that wasn't being done into a new area and shown productivity.
Alan, I know you want to respond to that.
No, I think if you think of it in a different kind of way, I mean, again, we have an opportunity to look from a very high level, and in where research activity is occurring, it's distressing. But then if you think about the science in any scientific area, the way you know you have understanding is if you can perturb it and predict the consequences. Well, the only time we actually start doing experiments in people where we're really testing medicine is in phase 2 proof of concept trials, and most of them fail. So at a completely objective layer, how are we doing? Maybe studying those 10% is the right 10%, we don't know. Well, the evidence suggests not, because when we go into people testing the completely new ideas for the first time, what's the efficacy rate? 60% fail in phase 2A of completely novel ideas. So we don't understand. So the repurposing efforts are based on existing knowledge of a system we don't understand. So it's going to be lucky if any of those repurposing ideas, I mean, ideas are cheap, right? You put 2 connect points, you can have ideas. But bluntly, the problem is not, the medicines, the tiny pharmacopoeia we have now. We don't understand human biology. Now we know why, because we're all focusing on 10%. And there's not going to be a big sort of step change in our discovery of medicines until we figure how to do more research on the, as a quotation marks, novel. There's sure going to be innovation in what we know already, but it just doesn't make sense from a purely high-level view of the world. It cannot be the source of new medicines.
I would respectfully take issue with this. I think there's a lot of merit in understanding basic biology and basic pathology, but it's also extremely complex, extremely expensive. It's going to take a lot of time. Some patients, don't have that time. The history of pharmacology is replete with examples of instances where we've thrown compounds at a disease and we've been lucky and we've had treatment decades before we could understand the underlying biology of the disease. So I think we need to follow both approaches. It clearly that latter approach that I'm describing doesn't work in all cases. I mean, you look at Alzheimer. We've done that at Alzheimer. We've thrown, you go to clinicaltrial.gov, over 200 compounds at Alzheimer, hoping that we'd be lucky, and we have not been lucky. So this is an example where maybe we need to rethink that approach and go to what Al is saying and trying to understand what caused Alzheimer before we spent more billions on that research. Because the reality is that if we knew today what caused Alzheimer, we'd probably have the technology within months to come up with something that's going to do some good. So we need to follow both approaches.
And if I could add, I think I fit perfectly in between the two of you. Elle had a great way to approach the problem and raise the question. And Bernard, exactly, how do we be practical about this? From the NCAT's perspective, a couple surprising things happened, I think. One is that we had an initial mission of going after rare and neglected diseases because no one else was doing it. And so the payoff there was, well, it's kind of limited, but these small groups of patients, it's huge, right? We can extend their lives. So that was the focus. And in doing that, though, what we ended up doing out of necessity is that we had to explore the unused biology, this unused space. And that was really sort of, at least to me, sort of a new, great discovery, new landscape, and it's paid off. And it's now extendable, so we now can investigate some of these rare neurological disorders. We now have new targets for studying Alzheimer's as a result. So there's great hope. The scientists are very excited, and now this extends to big pharma. So the big pharma model, you see, well, a lot of big pharma is now chasing after rare and neglected diseases. Well, you say, why are they doing that? Is there $1 billion in that? First, no, it's great PR, of course, that's as we expected, but also it's for the same reason. They're going after the new biology, and they're going to find these new target spaces that can be applicable, I think, to broad human health issues.
Yeah, it's a very tough question, Aled, that you've posed, because that's what you basically identified as an ecosystem problem, right? Because we would work on that other 90% if there was more money, and there isn't enough.
Or we spent the money differently. There's plenty of money. We don't need more money.
So, can I write that down?
There's 10,000 papers on MAP kinase. Do we need 10,000? Every single one. Average cost of paper is $200,000. Do the math.
Well, I think finding more money is easier than repurposing the money that's being spent right now, right? And so, I mean, all I can say is from the pharma point of view, is we'll invest where it makes sense. And so the rare diseases is one opportunity. I think what you said was, people are going there. Over half of my pipeline is rare disease, by the way. And we go there because the science is strongest there, right? And so I would argue that not every gene and not every protein is actually relevant to human health, right? And so maybe it's a combination of genetics and structural biology and medicinal chemistry that'll help us answer that question and help us prioritize what's relevant to disease. And that may not require any more funding, just more discussion. But certainly the rare diseases have shown us that genetics, human genetics, is very powerful and explains human biology to us, not just for the rare diseases, but related diseases, right? We do have the freedom to invest wherever we want to invest, right? And so I think maybe the private sector has the ability to do that. And we're allowed to fail because everybody expects us to. So, so we'll do what we can in that space. But I do think the ecosystem needs more resource. I really do. When every time we visit an academic institution in the United States, we hear how difficult it is to get funding to continue the work that's ongoing in this nation. That's a huge problem. There are other countries that are spending, absolute numbers are lower, but proportionately for their academic communities, more than we are. So that has to get fixed. That's a big problem.
Since we are in Washington, of course, we know that the problem is that it's probably not going to get fixed anytime soon. Yeah. And so what do we do given that? That's just a comment, not really a question, because I know we have lots of other questions. So Stephen, and then why don't we all go to the microphone because it's being webcast.
So Stephen Johnson from One Mind for Research. I think there were quite a number of anti-lawyer comments from the panel. But my question is, and really what it seemed to revolve around was the interest of academic collaborators in pursuing patents. So my question is, seeing as we're here in Washington, if Bayh-Dole was repealed tomorrow, would that increase or decrease the pace of innovation?
Is that a rhetorical question, Stephen?
It is a rhetorical question, yes.
Well, because there are a lot of people who are Bayh-Dole experts in the audience, I should note.
But it's an actual question.
Yeah,
which is,
yeah.
would it get rid of the roadblocks we've heard from the panelists where they said, and then it took 12 months, or then they want to file patents, so we couldn't do collaborations with universities in America, we had to do them in Canada or in England. That's the question.
I just have one comment from a practical standpoint. One of the it's a problem we have with Bayh-Dole in the government that most people outside the government don't have. That Bayh-Dole is sort of a it automatically applies in our collaborations. So if there's a contract for work and something gets discovered then, or if there's any sort of research collaboration, rights automatically vest. So if Bayh-Dole was pulled, then there wouldn't be that presumption, presumably, and maybe you could be able to put in some agreement language, collaboration agreement language that would preclude that from the beginning. There are ways to do it currently through a declaration of exceptional circumstances and whatnot, but that's much more administratively burdensome.
Bayh-Dole doesn't affect us because we collaborate with scientists, and there's no law that says you have to patent, right? The law says if you do patent, then this is what happens. So you have handshakes agreements with your friends. We agree never to patent. They don't patent, and it's not against the law. But we don't go through institutions because they don't understand that.
Yeah, I just want to say it's actually not the academics most of the time who want to patent. It's their institutions, right? Yeah.
So the negative comments are not technology transfer function, not
So my name is Josh Summer. I am from the Chordoma Foundation. Chordoma is one of these rare diseases that it sounds like everyone wants to work on these days. I'm also a chordoma patient. Chordoma is a rare form of bone cancer. So several years ago, we started the foundation. Virtually nothing was known about chordoma. Now we've learned a good deal about the biology of the disease. We've been dealt an interesting hand of cards in that some of the targets that seem to be relevant are in the kind of popular zone, and one of them happens to be out in the Canadian netherworld. But I wanted to follow up on a comment that Craig made, which is that the vast majority of meritorious trials or indications just never get pursued. And we're facing that situation right now where there is a tractable target with several approved drugs that seems to make a lot of sense in chordoma. There's compelling preclinical data, there's molecular data, but we haven't been able to we've approached a couple companies, we haven't been able to get anyone to bite to be willing to do a clinical trial or even provide drug for a clinical trial for chordoma. And so the foundation, we're trying to figure out how we go about financing this. Where do we find the money? That's challenging for us given our small budget, and so we've been thinking about maybe alternate ways that we could fund this sort of trial. One thing that came to mind is, would it be possible to attract private capital for a repurposing trial if there were some way to actually recoup some of the upside, if there were a supplemental NDA that were achieved with that trial? I don't know if that's ever been done before, but since we're in a room full of lawyers and we have people from the industry, I just wonder if that idea has been given any thought, if it's ever worked. This is very different than venture capital. It would be much more kind of project finance to incentivize more of these repurposing enabling trials.
Yeah, I can take a shot at that. I think there's exciting possibilities that are opening. One of the great things about the model that you've heard about it, drug repurposing and so forth, is that when you run the numbers on them, they eliminate up to 80% or above 80% of the cost compared to a traditional clinical trial. So what used to cost, let's say, $20 million, if you can do it a traditional way, you might be able to do it with $3 million if you do it, with an open source approach. And $3 million puts you in a range of crowdfunding approaches, which basically can fund up to $10 million, thanks to some recent legal changes. So that's another way why we, can embrace or should embrace those cheaper approaches, or at least give them a good look, because they can dramatically expand the range of research that can be done and funded economically.
So, Josh, the drugs in question are already generic for other uses, or are they
not generic? So still on
generic. They're still on patent for other uses, and so you would be able to get some additional exclusivity within the initial NCE patent life.
Exactly.
Well, that's very interesting. I mean, we should definitely talk, because I think that's a really good strategy.
Okay. Thank you.
I'm trying to decide whether I should jump in here or not, but it is all around a return on investment, and it depends on the stage of the compound, obviously. If the compound is already approved for another indication, then a publication may be sufficient in that new indication, particularly if it's a rare disease, to accomplish what you want to accomplish, which is get it to patients. If the drug is not yet approved, then somebody has to finance taking it through to get it approved, and that's where you have to come up with the business plan. It's either a foundation that does that, it's either the pharmaceutical company that does that, or it's a venture capital. The Patient Advocacy Foundation is the most generous in terms of what their tolerance on return on investment is. Both the pharmaceutical company and the venture capital ultimately are for-profit organizations, and consequently, how much they're going to invest, there's a multiple of that has to be on the return. And that's what inhibits innovation in the smaller diseases. And luckily, we're getting many companies, many private sectors that are now willing to invest in that area for the benefit of patients.
Dominique, did you want to add? Yeah.
Oh, great.
So Kaufman is facing exactly one of the same situations with Oronafin to some extent, and we should talk also about the possibility of publication, because that's an interesting possibility that I know that Dominique has considered as well. I'm not sure it's a panacea.
So on the point of publication, when there is a publication, presumably prescription patterns change, and maybe in the case of Chordoma, AstraZeneca, let's just say it's Aresa, would get another 500 patients a year taking that drug. Well, that would generate an increase in revenue. But I think in the absence of supplemental NDA, I don't know how one would track or participate or somehow determine what additional revenue was gained and allocate that or somehow return that to investors. So in a way, I think that could be a solution, but we have to work through specifics as to how that investors could participate in that upside.
One other aspect of the IP picture is not just patents, but it's also the diagnostics behind it, which are increasingly being less patentable. Having a special patient group like yourselves, I don't know what the diagnostic situation is for your group, but you can leverage that certainly in this unused biology space, right? So you bring to the table a special subset. You have your patients, and there's a special biology that will give clues to potentially bigger problems that can be solved.
Jerry.
Jerry Reichman, Duke Law School. Thank you for these inspiring presentations. It was particularly gratifying to see the opening of the compound libraries, an approach that Arti and I wrote about several years ago in an article. I would like to know about one concern that we had in opening up the libraries, putting ourselves in the shoes of the possessors for a moment, the risk of trade secret theft and industrial espionage on the state of research going on that might prove useful to other companies. Of course, if everybody's failing, there's less value to steal. But I do wonder if that is still a concern, or if you have found ways in your standard template agreements to deal with that satisfactorily.
So it is a huge concern, and every conversation that we have within our company, that's always a pushback. Well, we don't want to disclose this yet, or we're not ready to disclose that, or it can give our competitor an edge if they knew if our compound had this wart or that wart. We need to get past that. Mm-hmm. For several reasons, but mostly because that compound isn't ever going to not have that wart. So letting it known isn't going to change, isn't going to make that compound better. And if you're making another molecule that is better than that, you already know that, and you're already years ahead of somebody else figuring that out. So as we've had the conversations, we've become much more comfortable with we can disclose the properties of the compound. And now it's a matter of, you know, how do we succinctly, what information is important for the investigator to know? Because it's not easy to open up everything to the investigator. It's more what they need to know to be able to do their particular
Well,
study.
that's really great news. That's one of the things we hope would happen, that you would have an internal a committee.
Debate about it.
So relatedly, and I know Don Farrell has spoken about this, it's expensive though sometimes to gather up all that old data.
It is.
And you know, to what extent is that the bigger impediment now? Because you know, I think Alad was, I don't know, Bernard was saying, why not open up your vaults of
Right.
all your failed compounds? And it sounds like it's expensive to open up your vaults even if you wanted to.
So I need to comment on that and correct one of the things that Bernard said. There aren't thousands of compounds sitting on drug company shelves that are patient-ready. There were thousands of compounds that were brought into clinical trials, but many of those failed to hit the target or to be tolerable, sufficient to move to patients. So,
Will you reveal the
many of
negative
them we
results?
don't even know whether they hit the target strongly or drove them to doses where they were tolerated or not. So we've went back through. I came from Pfizer just over a year ago. And Pfizer, being the largest pharmaceutical company, has more failures than any other company, which gave us a lot of opportunity to understand why we failed, but also a lot of compounds that we could go back to. And we went through hundreds of compounds, about 500 different compounds against about 250 different mechanisms. And we ended up with, on the order of 30, that we had good confidence hit the clinical target in man and were tolerated. Done the same thing with AstraZeneca, and we're about at 16 or 17. These are of discontinued compounds. So there isn't a large number of compounds out there. Now, across the industry, that expands to 58 when you do an NIH call for proposal. And, of course, there's new molecules that are being generated every day. But it isn't large numbers. Now, to address your question, sorry for taking so long, but going back to discontinued compounds, it is difficult to dig out that data. And in many cases, the data doesn't actually exist on target coverage, so you aren't able to include the molecule because you just aren't comfortable to be able to stand in front of an investigator and say, yes, you can test your hypothesis because this molecule will hit the target and allow you to test your hypothesis.
So everyone wants to speak, but I'll let Bernard.
Yeah, let me push back a little bit on that. I do believe there is thousands of compounds out there. Now, they may not be all patient-ready and they may be various stages of development and may have failed, but I remember I was sitting next to a big pharma CEO in April of last year when these 38 compounds, or 58, whatever the number was, that list was published. And I remember that one of the brightest, most effective CEO in big pharma, for whom I have a lot of respect. And that person shook his head saying, we made those compounds available. Frankly, I don't understand why the interest and what those scientists, some of the scientists might be able to find. If there was some value in those compounds, we would have found it ourselves. Without realizing that maybe you try this compound in oncology, but you could try it in, in psoriasis at 1/10 of the dose, and what might have been toxic in oncology might not have been toxic, or maybe you can try a topical route administration as opposed to injectable or whatever. There's multiple reasons why something that has failed at some point and with seemingly no future could be usefully repurposed for entirely. And in fact, the experience of 58 compound just proved that. So that's I really believe that if the compounds, I mean, you go to clinicaltrials.gov and you get thousands of compounds there, many of whom have been kind of fallen by the wayside. You don't even know if they're active or not. They're just there. They've been sitting there for 10 years. So you got to presume that they're dead. Those compounds could be doing some good if they were offered to the general public or the scientific community saying, hey, these are standard terms. You can access that if you, can be imaginative enough to find a use for it.
I want to get back to your concern and your question about industrial espionage because, and I want to go back to this early space because, we spend a lot of time and a lot of effort, a lot of money creating these chemical libraries that we hope will represent diverse chemistry, diverse chemical space. But to invent a medicine from that initial screen takes, somebody said 18 months of medicinal chemistry. I've been on programs that have taken 4 or 5 years. So the number of inventions that you have to do beyond that initial screen is enormous. And so in terms of industrial espionage, there isn't a single medicinal chemistry program in this industry where people aren't closely monitoring their competitors' IP. And as soon as structures are published, even if they're not explicitly stated in a patent, our chemists are good enough so they can usually figure out, what the 2, 3 best compounds are, and they go and they make them and they start testing them, and everybody starts to reoptimize. So, that's legal espionage, and it's an 18-month delay really from, the filing to when it comes out. So there's a huge amount of information that's being traded between the companies, whether it's intentional or not. And then this gap after you run a screen and you get a hit is so big that there's so much more room for invention. So, the diseases of the developing world published, I think it was tuberculosis, they ran a screen, they published all the hits. They said, okay, everybody go out and pick a hit and go work on it. I mean, the other thing we see is that we'll run a screen, we'll get 10,000 hits, 1,000 hits, we'll pick the 100 best ones. You pick, we have a saying, if you have 10 medicinal chemists, they'll each pick a different compound because, everybody's skill set and interests and passion is around a certain, methyl group or a certain pyrimidine or a certain, so people, different scientists will see things differently, and that's actually what you want, right? Because at this early stage, you don't know what's going to happen. So that's how we do it within an organization. Can you do it by including other organizations as well? So in terms of Discovery Fast Track, part of our MTA was divorce terms. If we decided not to work together after a screen, we actually asked the academic to publish the hits. Right, get them out there. If they want to go continue invent, if we want to continue invent, if Pfizer wants to go or AstraZeneca wants to continue invent, I say go at it because the failure rates are so high, right? I mean, you don't want to protect your idea to the point where you squelch the innovation and the ability to move to the next point. Sometimes you just have to let it go.
Yeah, just one more word. I think your concern is very real. It's omnipresent across the industry. With all due respect, I think it's way overblown because there is overwhelming evidence from research that the cross-pollination between scientists and the bold thinking that you get from cross-pollination are 2 essential ingredients of innovation. You need that in order to get innovation. If you don't have it, which is what you eliminate when you start hiding things, you get what we have today in the industry, an industry-wide innovation crisis, innovation failure.
Colvis, do you want to add something else?
Nope, agreed.
Yeah, I just wanted to, that's okay, I just wanted to add, looking at the other spectrum, so we've talked here about repurposing or redirecting shelved compounds. I'd like to just put this question out there to people in the industry especially. Well, what about the live compounds that are making you profit now? Certainly there's a risk for you to start these secondary clinical trials for second purpose, and if there's an adverse event with this other patient population group, that seems to be a huge risk, and there's a reason that you might not want to do that. So putting it out there then, is there, and looking forward, your live compounds will become generic at some point, and there's potential for off-label use then. Is there a strategy for repurposing or redirecting somewhere in their lifetime?
So the compounds that are already on the market are already out there. The investigators know about them. They can submit proposals to the pharmaceutical company to do trials in new areas. They can submit proposals to funding agencies and to do those. So those are already out there. So what you're really asking is around the compounds that are in early clinical development. And there's been lots of discussion about those as well. Again, those have been hands-off in the past, and they shouldn't be. And that's really the best time from a patent point of view, from a knowledge point of view, from a freshness of the target point of view. These are only 10 years old rather than 20 years old from the identification of the target, and we are starting to make those available at this point. We did in our Taiwan relationship that was just announced a month ago. There were 4 live compounds that were put into that program.
Is there just a general number of which are in that category compared to all your compounds? What percentage? Well,
you can go on websites for each of the pharmaceutical companies and you can see their active portfolio. There are smaller numbers than those that have died in the past. So yes. And of course, you still want to get them to the stage where you know that they've hit targets, so you know the hypothesis can be tested. Companies get very funny when you get into phase 3 or close to registration, because they're very worried that anything that comes up in an ancillary study that the regulatory agencies, the FDA, is going to zero in on and delay the process of registration. And I just don't think we give the FDA enough credit.
It's been a long time since I've heard the FDA praised. So on that positive note, let's break for lunch. I know that I'm sure folks will have lots of questions for the panelists during lunch. Oh, you had one? Very quickly, because we only have 45 minutes for lunch. This is a jam-packed program.
Okay. I'm from NIH, NIDDK, and I want to thank you again for a very informative presentation. We actually work on quite a few collaborations with industry to do the large phase 2 multi-center clinical trials, particularly with active compounds. And we do it through our consortium. And one of the questions, or one of the barriers that we often face is particularly for active compounds that are close to a phase 3 trial for a particular indication that are being tested for a different one. There's some reticence by industry to share data until, and this is I can understand appropriate regulatory strategy to share data and have it in the public domain until after they've actually filed or initiated their phase 3 trial. And it goes back again to their concerns that if there's an adverse event that comes up in an ancillary study, that it may delay their approval. And I think that what's often not taken into account and not credited is the FDA, because they're looking at a vast patient population. So we're sort of grappling with what we can do at the NIH to try and assuage some of the concerns and have industry partner for some of those trials where we're looking at a different indication. And it would be helpful to get your insight on that.
Very quickly, continue to push and continue to have the conversation. I think the industry is at the stage of breaking ground on that and opening up. And so don't take your foot off the pedal.
More positive notes. Okay, lunchtime. Lunch is in the, what's that? Oh, thank you to our panelists, of course.